We hope you have a great day, and we hope you listen to and take in what matters most to patients in this guidance. Most recently released by the FDA about patient-focused drug development and read by different patient advocates, all 39 pages. We hope you enjoy it. Thanks for listening. Thanks for all you do. Any last words?
I hope you have a great day and think about other people and your friends.
Hi, I'm a rare disease patient advocate and caregiver, and this is important. In February of 2022, the FDA released a new guidance for industry. It's called Patient-Focused Drug Development, Methods to Identify What is Important to Patients. It's so important that a bunch of my friends have gotten together to make it easy for you. We are all going to read every page of this 39-page report to you. That's how important it is. So we hope you will sit back and listen to this important guidance from the FDA. And thank you so much for working with patients and families to design the best possible treatments. For the purposes of this report, the recording will not include footnotes, but you can find the report online to learn more. Thank you.
Page number 1. Patient-focused drug development. Methods to identify what is important to patients, guidance for industry, Food and Drug Administration staff, and other stakeholders. This guidance represents the current thinking of the Food and Drug Administration, FDA or agency, on this topic. It does not establish any rights for any person and is not binding on FDA or the public. You can use an alternative approach if it satisfies the requirements of the applicable statutes and regulations. To discuss an alternative approach, contact FDA staff responsible for this guidance as listed on the title page. Number 1, introduction. A, overview of the series of FDA guidance documents on patient-focused drug development. This guidance, Guidance 2, is the second in a series of methodological patient-focused drug development, or PFDD, guidance documents that describe how stakeholders, patients, researchers, medical product developers, and others can collect and submit patient experience data and other relevant information from patients and caregivers to be used for medical product development and regulatory decision-making. The topics that each guidance document addresses are described below. Methods to collect patient experience data that are accurate and representative of the intended patient population, guidance number 1. Approaches to identifying what is most important to patients with respect to their experience as it relates to burden of disease, condition, and burden of treatment, guidance number 2. Approaches to selecting, modifying, developing, and validating clinical outcome assessments to measure outcomes of importance to patients in clinical trials, guidance number 3.
Methods, standards, and technologies for collecting and analyzing clinical outcome assessments data for regulatory decision-making, including selecting the COA-based endpoint and determining clinically meaningful change in that endpoint. Please refer to Guidance 1 and other FDA guidances for additional information on patient experience data. In conducting research that involves accessing patient experience data or directly engaging with patients, it is important to carefully consider federal, state, and local laws and institutional policies for protecting human subjects and reporting adverse events. For additional information about human subjects protection, refer to Section 4A2 of Guidance 1. FDA encourages stakeholders to interact early with FDA and obtain feedback from the relevant FDA review division when considering collection of patient experience data related to the burden of disease and treatment. FDA recommends that stakeholders engage with patients and other appropriate subject matter experts, for example, qualitative researchers, clinical and disease experts, survey methodologists, statisticians, psychometricians, patient reference researchers, when designing and implementing studies to evaluate the burden of disease and treatment and perspectives on treatment, benefits, and risks. Purpose and scope of Guidance 2. This guidance will discuss methods for eliciting information from individuals identified in Guidance 1. It will discuss best practices in conducting qualitative research and reference-related resources. However, it should not be viewed as providing detailed instructions on how to use particular methods or as a substitute for engaging subject matter experts when undertaking the work described. The methods described in this document can be used to elicit what is important to patients, which may in turn help inform understanding of disease condition and clinical trial design. It may also help the generation and use of patient experience data, including clinical outcome assessments and patient preference information, to inform benefit risk assessment.
This guidance does not address methods for collecting and analyzing COA data, a topic to be covered in later guidances in this series. It also does not address methods for collecting and analyzing patient preference information, which is addressed in other FDA guidances. The contents of this document do not have any force and effect of law and are not meant to bind to the public in any way unless specifically incorporated into a contract. This document is intended only to provide clarity to the public regarding existing requirements under the law. FDA guidance documents, including this guidance, should be viewed only as recommendations unless specific regulatory or statutory requirements are cited. The use of the word should in an agency guidance means that something is suggested or recommended, but not required. Methods to identify and understand what is important to patients. A, background research. Research to understand what matters most to patients living with a disease or condition to guide medical product development should begin with a characterization of the disease or condition and currently available therapies. Before conducting studies in patients, which may involve their caregivers as well, literature reviews, consultations with relevant subject matter experts, and other information sources should be used to develop target research questions and select appropriate methods to identify what matters most to patients regarding their experience with their disease or condition. B, overview of methods. When planning research, consider whether the study sample reflects the group of individuals, patients, of interest, for example, the target population, including the spectrum of disease or condition and patient diversity, and whether the methods used to elicit information from patients are appropriate for the research objective and target population. Qualitative research methods, quantitative research methods, or mixed method research can be used to identify what is most important to patients. These methods can be used either independently or complementarily. When selecting an appropriate research method or set of methods, FDA recommends carefully considering the research objectives. Qualitative research methods, such as interviews and focus groups, are typically used to obtain a deeper understanding of the patient experience by generating in-depth information about the experiences, perspectives, priorities, preferences, and feelings of patients and others in their own words. Quantitative research methods, such as surveys, are characterized by the collection of quantifiable data, for example, numerical data, and the application of statistical methods to summarize collected patient experience data, to describe, compare, or relate measures of patient experience. Mixed method research, such as survey instruments with open-ended and fixed response questions or interviews combined with administration of survey instrument with fixed response questions, involved using both qualitative and quantitative approaches or methods in a single study or program of inquiry to understand the patient experience.
Quantitative research methods, such as surveys, are characterized by the collection of quantifiable data, for example, numerical data, and the application of statistical methods to summarize the collected patient experience data to describe, compare, or relate measures of patient experience. Mixed methods research, such as survey instrument with open-ended and fixed response questions or interviews combined with administration of a survey instrument with fixed response questions, involves using both qualitative and quantitative approaches or methods in a single study or program of inquiry to understand the patient experience. Although this document includes distinct sections for qualitative and quantitative research methods, many data collection methods may be used in either approach. For example, interviews are commonly used to generate qualitative data, but they also may be used to generate quantitative data. Similarly, survey instruments are commonly used to generate quantitative data, but may also be used to generate qualitative data with open-ended questions. Part 3. Qualitative research methods. A. Common qualitative methods used to obtain patient input. One-on-one interviews and focus groups are two common qualitative methods which will be discussed in this section. Other commonly used qualitative methods are summarized in Appendix 1. There's no single preferred qualitative method for all uses in research questions. The method selection should be suitable for the research objectives and questions. FDA will review the rationale and appropriateness of the qualitative method chosen. Before selecting qualitative data collection methods, consider patient characteristics and the potential strengths and limitations of the methods discussed in Table 4 in Appendix 2. There also is not a universal number of interviews and focus groups to conduct. It is dependent on the research objective. For certain methods, it may be guided by the achievement of saturation. See Appendix 4. 1. One-on-one interviews. One-on-one interviews, for example, one person interviewing another person, involve a discussion on the topic of interest between the study participant and a trained interviewer. One-on-one interviews offer opportunities to explore topics in depth at an individual level using probing questions. There are 3 types of interview methods. Structured interviews. The interviewer asks a set of predefined questions. Semi-structured interviews. The interviewer asks a set of predefined questions and probing questions. And unstructured interviews. Interviewer asks unplanned or spontaneous questions. When using one-on-one interviews, it's important to consider the following: patient selection and sample size, selecting sources for participant recruitment, for example, medical practices, centers, academic institutions, research consortia, polling organizations, national panels, where applicable to recruit participants for in-person interviews. Choosing an appropriate sampling method, refer to Guidance 1. Estimating an adequate number of interviews to conduct while considering number of sites, as well as geographic and patient representation. A greater number may be needed for unstructured interviews, broad or complex topics, or diverse populations. Interview and data collection methods. Selecting the appropriate interview type and administration method, for example, in person or remote, for the target population. See Section 3, 8.3 for more information on administrative methods. Designing suitable interview questions and interview guide. Focus on concepts for importance for context of use and research objectives. Pilot testing interview guide, and administer the interview in a small number of participants to identify and correlate any methodological or logistical issues before using in the quantitative study. Interview conduct. Pilot testing length of interview and appropriateness of length for administration method. Selecting and training interviewers to perform interviews, for example, considering expertise in performing interviews and other factors based on characteristics of the disease or condition and target population under study. The emotional burden for the respondent. Potential for heightened emotions, including anxieties and discomfort among patients and caregivers, as well as the emotional burden for the interviewer. Potential for emotional distress associated with hearing about difficult patient and caregiver experiences may affect responses. 2. Focus groups. Focus groups involve a conversation with a group of participants led by a moderator. The moderator can explore issues at the individual level, as well as encourage discussions among participants at the group level. This approach elicits a range of experiences. There are different types of focus groups, however. The most common is a single focus group, an interactive discussion of a topic by a set of participants and a moderator as a group in one place. Generally, a moderator uses semi-structured design discussion guide to direct the group conversation. The considerations for focus groups are similar to one-on-one interviews. Choosing an appropriate administration method and sampling method, designing suitable questions and discussion guide, pilot testing the discussion guide, and the length of meeting. Other important considerations for focus group include selecting a trained moderator with the appropriate skills, choosing the number of focus groups to conduct, which may vary based on factors such as complexity of topics, for example, all versus some impacts of a disease or condition on multiple dimensions of a patient's quality of life, dependent on therapeutic areas and research questions. Sensitivity of the topic, for example, separation of genders for sensitive topics such as sexual function, diversity of the participant sample, number of the subgroups planned, for example, different age groups, disease or condition severity groups, gender. Determining the sample size for each focus group. Generally, the goal is to keep groups small enough to enable the elicitation of an in-depth responses from each participant, but large enough to get a wide variety of perspectives across different severity levels and demographic representation with the target disease or condition. Although there is no set number recommended for a focus group sample, sample sizes between 5 and 10 participants are common. A group may become fragmented. For example, multiple simultaneous conversations occur when there are too many participants, decreasing the likelihood of engagement and responses from each individual. Number 3, choosing between one-on-one interviews and focus groups, in person or remote. Both one-on-one interviews and focus groups may be useful to capture patient experience data. For example, focus groups could be used in preliminary research to explore a broad topic, followed by one-on-one interviews to obtain more detailed information on the topic of interest. Each interview method has its own strengths and limitations. One-on-one interviews can be used to obtain more detailed, in-depth individual experiences, address sensitive topics, and explore disease or conditions with many different symptoms that vary from patient to patient. For example, heterogeneous disease, condition, presentation. Focus groups allow for participant interaction and can capture a range of perspectives with the population in a shorter time frame. One-on-one interviews and focus groups can be conducted in person or remotely. For example, use of computers or telephone. FDA does not have a single recommendation administration method for conducting interviews and focus groups. However, the method should be appropriate for the selected target populations, study characteristics, and study objectives. FDA will review the rationale and appropriateness of the administration method chosen for the intended use.
Each administration method has strengths and potential limitations. A growing body of literature suggests that there are no marked differences between interview administration methods regarding the accuracy of the data collected. Table 1 on page 7 lists some potential strengths and limitations for in-person and remote administration methods. Some strengths and limitations are not entirely distinct for the methods, which include in-person, telephone, audio only, and online virtual video conference. Approaches to asking the right question. Regardless of method, the way questions are framed is critical to collecting unbiased patient input. Although spontaneous responses are ideal, there are situations in which participants may need to be prompted. Prompts, i.e., open-ended questions to stimulate and provoke participation in the discussion, are used to help the interviewer moderator gain more information, particularly if the participant does not initially provide detailed responses. Prompt questions should avoid leading the participant. Leading questions, i.e., questions that include or imply the desired answer to the question in the question, are problematic because they may result in biased or false, misleading results. They may also lead to a missed opportunity to hear an unexpected insight. Approaches to avoid leading questions include, but are not limited to, do not suggest an answer. Do not assume you know what the participant is thinking or feeling. Do not ask questions that cast judgment on a participant's belief, choice, or perspective, or imply that you prefer that the participant to respond in one way versus another. The box text that follows on page 8 offers some examples of probing questions or prompts that are leading or otherwise problematic and offers potential solutions. Example 1, research objective. Determine what aspects of peripheral artery disease patients would like to see improved with treatment. Leading or probing question. Wouldn't you consider it most important to improve your walking distance? For example, how far you walk around the track when you exercise. Problem. This question guides the respondent to provide an answer that is more favorable or preferred by the researcher. Additionally, the for example clause may not include relevant examples for the research participant. Potential solution. Consider rephrasing as, tell me how peripheral artery disease impacts you. What would you like to see improve with treatment?
Section 4, Quantitative Research Methods. Survey research methods are commonly used to collect quantitative data from patients and relevant stakeholders. Refer to Guidance 1 for considerations for data management and data analysis. In designing a survey instrument, it is important to decide how to administer the survey instrument and how to design and test the instruction questions and response options. Section A, the choice of survey administration method. Survey instruments can be self-administered or interviewer-administered. Self-administered surveys can be paper-based, telephone-based, or electronic-based. Interviewer-administrator surveys can be conducted in person or remotely. Choice of administration methods may be driven by a variety of factors. For example, an interviewer-administrative survey instrument, a survey instrument using an interactive voice response system, or a survey instrument easily completed using common assisted technology on a computer may be useful in patients with visual impairment. Self-administration allows participants to respond at their own pace and at their own convenience. Compared to interviewer-administrative surveys, self-administration typically is less costly and removes the potential for interviewer bias. Computer-assisted and administrative survey instruments can navigate skip patterns, help minimize missing data, and allow real-time data collection and analysis compared with other methods. Administering survey instrument in a clinical trial at screening or an exit visits or after the end of the treatment may add greater depth to understanding the burden of the disease or the condition, the treatment, and trial participation, as well as provide more detail on patients' perspective on the treatment benefits, the harms, which may help inform drug development and COA development.
Considerations for developing questions and response options for a survey instrument. Survey instrument instructions and questions should be well aligned with the research objectives and designed to answer the research questions, specific to the targeted concept for the research question. For example, disease, conditions, symptoms, and impacts, current treatment, past treatments, and treatment side effects, with minimal redundancies across questions. Well understood by participants to enhance consistency of response, including having been assessed for appropriateness for the ability of the population. For example, reading and writing ability or literacy, ability to understand health information, health literacy, and the ability to understand and use numbers, numeracy. developed to include natural, familiar language. For example, minimal use of clinical terminology, as used by patients when discussing the concept of interest. Assessed for translatability of questions if used in multinational and multicultural studies. Ultimately, the survey instrument should be translated and culturally adapted for all languages and cultures where it will be administered. Assessed for applicability of the content, although sometimes a not applicable response option is also needed for a question. Tested through interviews of respondents in the target population to make sure they interpret the survey instrument's instructions, questions, and responses as intended and can respond accordingly, including that question stems and response options are appropriate and meaningful. Alternatively, tested through the survey instrument, for example, inclusion of survey questions to measure comprehension of attributes, questions, and concepts in a format that can be analyzed so that comprehension is part of the main analysis. Formatted in a simple manner to maximize the ease of use for respondents and interviewers. Assessed for potential response bias. For example, the tendency of respondents to answer questions in a manner they perceive may be viewed favorably by others. Tested to ensure electronic or web-based technology is functional and or compatible to administer the survey instrument, if applicable. Scripted to ensure standardization if administered by an interviewer. The FDA discourages the use of incomplete questions, for example, age, reason you last saw the doctor, leading questions, unclear or confusing questions, for example, poorly worded questions, double-barreled or multi-barreled questions, for example, a question that asks about two or more concepts at once, double negatives, for example, a sentence that includes two negatives. When developing questions, consider the respondent effort to complete the survey instrument because this effort can contribute to respondent burden, which can result in a lower quality data and non-response. Minimize respondent burden by including a simple survey design and layout, including simple questions, non-repetitive questions, pre-testing of questions, and time testing, estimate to complete the survey. Examples can be found in the table on page 11. There are two types of questions that can be used in survey instruments: close-ended questions, questions with a fixed set of response options, and open-ended questions, questions without a fixed set of response options, for instance, free text. Table 2 on page 11 has lists of examples of closed and open-ended questions, as well as some strengths and potential limitations of using both types of questions. Table 3 examines some different types of response options. Note that this is not an exhaustive list. Please review Table 3 on page 12.
The ordering of questions in a survey instrument may also be important. Priming occurs when information presented in an earlier question causes respondents to adjust their responses to subsequent questions. Ways to avoid priming include the ordering and spacing of questions. The order of questions generally should flow from general to more specific to avoid order bias. Appropriate spacing of questions, for example, specific topics on different pages or electronic screens, can also minimize biased response. The use of screening questions can result in priming. However, in some instances, a screening question may also be useful to ensure that the survey instrument is appropriate and relevant to the respondent. For example, a survey instrument has been used, For example, a survey instrument has been designed to assess the burden of using a colostomy bag or stoma bag. A screening question would be useful to avoid including survey respondents in which this subject matter may not be of relevance. For example, the question might be, do you currently use a stoma bag? Yes or no? 5. Mixed methods. Studies using mixed methods research can use a sequential or concurrent design to explore or confirm a concept of interest. Example 1. Mixed method study based on qualitatively driven sequential design. A qualitative study uses focus groups to gain broad insight into patients living with diabetes. Following the focus groups, one-on-one interviews are conducted in a different set of individuals to attain a deeper level of understanding of the experiences and feelings of patients with diabetes. The information from this qualitative research, i.e. focus groups and one-on-one interviews, is subsequently used to develop a diabetes-specific survey instrument to better understand the prevalence of the generated concepts in a larger patient population. Example 2. Mixed method study based on a qualitatively driven sequential design. A study explores depression and anxiety in patients with acute coronary syndrome by administering a survey instrument to patients with ACS, a quantitative component. Analyzing the survey data, researchers find an association between depression and anxiety for female patients with ACS. In the second phase of the study, the researchers conduct follow-up qualitative interviews with a sample of the patients, targeting depressed male and female patients with ACS, to explore why the relationship appeared to be present only for female patients. Qualitative component. Example 3. Mixed method study based on concurrent design. A study examines the patient experience living with ALS, right? A study examines the patient experience living with ALS through one-on-one study interviews with patients and caregivers. This is the qualitative component. Within the study, there is concurrent collection of symptom checklist data, qualitative component. The data from both study components are analyzed separately before being compared. All right, page 16. This is a new sentence, but I'll read it anyway. FDA encourages researchers to consider the goals and objectives of using a mixed methods research approach and how the results from both qualitative and quantitative research components are intended to be used together. Thank you.
FDA encourages researchers to consider the goals and objectives of using a mixed methods research approach and how the results from both qualitative and quantitative research components are intended to be used together. Reasons to use a mixed methods research design may include harmonizing and confirming results from different methods, triangulation, supplementing and clarifying results from one method with results from another method, complementary, using results from one method to inform the design of another method, discovering inconsistencies, contradictions, and new perspectives, and reframing of questions or results from one method with questions or results from the other method, initiation, expanding the scope, range of a research question by using different methods for different components of the research question, expansion. Questions researchers should ask to determine which methods research approach to use to address their research questions. One, will qualitative or quantitative methods be more predominant in the study, or will both be given equal status, i.e. equal weight or equal priority in the study? Two, should qualitative and quantitative components be carried out concurrently or sequentially? With a mixed methods research approach, there's a possibility that the findings from the qualitative and quantitative components may appear to conflict with each other. FDA encourages researchers to consider approaches that increase the understanding and interpretation of potentially conflicting findings. The selected approach should be guided by both the context and the research question. 6, managing barriers to self-report. Typical techniques and/or methods to obtain self-report data may not be appropriate for all target populations and may need to be tailored to the population of interest. Consider the following approaches to obtain self-report data across broad populations. 1, participants with different capabilities, e.g. physical, sensory, intellectual, and/or communication. And within that, ensure usability of materials by participants with varying abilities or aids, e.g. low vision, tremor, using mobility aids. Bullet, use written materials with large or adjustable font and that are screen reader accessible for respondents who are visually impaired. Offer materials that can be read with a screen reader or in an interview format for those who cannot read. Use instruction modification, infrared eye trackers, and computerized tasks for people who are minimally verbal or nonverbal. Second bullet, evaluation of the capabilities of people with intellectual disabilities through appropriate procedures or measurements to ensure that they can validly provide their direct insights. Subpoint, consider certain populations may have a limited attention span, e.g. young children. Second point, plan to conduct interviews in shorter segments. Pediatric populations, ask young children to participate in drawing activities to help elicit concepts. Second point, use props. Populations with caregivers. For respondents who have a caregiver, the caregiver generally should not be present during a patient interview, e.g. they may be asked to sit outside the room. In cases where it is important for the caregiver to be present with the patient, e.g. for patient comfort, the caregiver should sit behind the respondent to minimize influencing the interview, either verbally or non-verbally. In either case, the research protocol should include a clear plan to document the presence and or assistance of a caregiver and how data from the patient and caregiver will be collected and reported in these instances so that the source of information is clear for analysis and reporting. For patients who are unable to self-report, eliciting what behaviors caregivers observe in the patients, including things the patients tell them, can help to avoid proxy reporting, i.e. reporting from the caregiver as if they were the patient. Proxy reporting can lead to inappropriate interferences and may not be reflective of what a patient may be truly thinking or feeling. Sometimes the barrier to self-report is language, dialect, or culture. Consider the following. First bullet. Questions used in multicultural qualitative and quantitative studies should be culturally sensitive and use language that is adapted to the culture of interest to ensure appropriate responses. Second bullet, it is important to understand and consider how cultural differences may affect participants' responses. Third bullet, poorly translated survey instruments can prevent researchers from collecting data comparable to that of surveys in the source or original language. Ideally, translatability assessment should be performed early during development of a survey instrument to address the needs of different nationalities, regions, and cultures. Fourth bullet, in both qualitative and quantitative studies, translation and cultural adaptation procedures for multinational, multi-regional, and or multicultural survey studies should be used to keep the meaning of questions similar. Fifth bullet, In survey instruments, it is generally helpful to keep the format of the questions similar across translations, considering the differences among languages, and to retain the properties of the instrument, such as range of response options and scoring. 7, considerations for use of social media. Considerations for use of social media. Social media may be an approach to collect qualitative and/or quantitative data. First bullet, data can be collected qualitatively through a passive observation of social media discussions or information. Observations can occur retrospectively or prospectively. Second bullet, data can be collected prospectively by administering a survey instrument in a social media setting. Best practices for designing and implementing studies using survey instruments and technology also are applicable to the use of social media to conduct a survey. Consider the following when using social media data. First bullet, choose an appropriate research design. First subpoint, mixed methods, sequential research designs can further strengthen the depth of knowledge gained from social media data. Second bullet, carefully select the sources of the social media with the research question in mind, because findings across social platforms may be distinctly different, e.g., certain platforms may have strong advocacy, support, community presence. Others may predominantly capture industry, academic perspectives surrounding certain issues. First subpoint, different social media communities appeal to different segments of the population, and the degree of a community's user anonymity may affect what users are willing to discuss. Research using social media data should examine a variety of social media networks and communities to obtain data that can be most generalized to the population of interest to the extent possible, i.e. generalizability. A discussion of the strengths and limitations of using social media data in qualitative, quantitative, and mixed methods research can be found in Guidance 1. Second bullet, third bullet, using appropriate methods to collect and analyze data. First subpoint, data collection methods should address potential limitations, e.g. lacking mechanisms to verify patient characteristics such as identity or diagnosis, and how these limitations can affect data integrity and interpretation. Second bullet, assess data quality. First subpoint, verify content and sources to avoid false information, e.g. bots. Third bullet, protect privacy. First subpoint, although verified patient community data may be used, in some cases it may be appropriate to allow users to remain anonymous or post under a username, e.g. blogs and forums, particularly when topics are of a sensitive nature.
Appendix 1, Other Qualitative Methods. In addition to one-on-one interviews and focus groups, refer to Section 3A. There are other qualitative methods that can be used to elicit what is important to patients, some of which are described in the following sections. A, Delphi Methods. The Delphi panel technique is a multi-stage survey process with the intent to achieve consensus among experts, which may include patients on a topic or issue. B, Observational Methods. Observational research methods can involve observations of patients by the researcher in a naturalistic setting. Example, home or school, a research facility, or virtual environment. Example, online communities, social media, to generate data related to symptoms or daily life functioning. These methods often involve assessment of events, patient attitudes, and behaviors over a period of time. Observational methods might be used to help understand experiences described through other methods. Examples of scenarios where these methods could be useful include, but are not limited to, in-person observations of children with attention deficit hyperactivity disorder in a classroom setting. Room surveillance live or through use of video recording to capture behavior while sleeping. Room surveillance for observation of aggressive behaviors or confusion in patients with advanced Alzheimer's disease. Social media listening. Example, observing interactions among social media users in an online community to understand how patients with a disease or condition describe their experience with the treatment. C, facilitated discussions at patient meetings. Facilitated discussions in well-organized public meetings that include patients, caregivers, and patient representatives can generate useful public input and patient perspectives in specific disease condition areas or topics. FDA has organized and led such meetings under the PFDD initiative. FDA also welcomes patient organizations to identify and organize patient-focused collaborations to generate public input on the disease condition areas, using the process established by FDA-led PFDD meetings as a model. D. Survey instruments with open-ended question, Section 4. Contains non-binding recommendations. Appendix 2. Considerations for selection of qualitative data collection methods. Table 4. Potential strength and limitations of qualitative data collection methods. Data collection method. One-on-one interviews. The strengths. Can gain in-depth and broad information on the topic of interest, include nuanced data about individuals' experience and perspective. Can gain an understanding of how a respondent interprets questions that might be included in a questionnaire. Flexible format can tailor interviews to generate appropriately detailed information based on research needs, example, through use of probing questions. Greater scheduling flexibility compared with focus groups. Privacy and confidentiality, some people may be reluctant to share certain things in a group setting. Can be conducted in person, at a study site, or at a person's home, or via telephone or video conference. Limitations. Time intensive. Example, length of time it takes to conduct several patient interviews. Participants may be uncomfortable providing complete or truthful information on sensitive topics to interviewers in person. Studies can be expensive. Example, staff time for conducting multiple interviews and data analysis. Focus groups. Strengths. Can gain in-depth information on the topics of interest as a whole. Flexible format. See above for one-on-one interviews. Efficiency. Elicit feedback from multiple participants at one time. Participants can react to and build on each other's ideas. Relatively inexpensive. Limitations. May not be efficient in covering maximum depth on an issue for each individual. Participants may become distracted by other participants in the group. Participants may experience peer pressure within the group. Single individuals might dominate the conversation, preventing multiple perspectives from being shared. Group settings may inhibit some individuals from providing sensitive information. Participants may be uncomfortable with speaking in a group setting and/or a public setting. Less flexibility in scheduling for a group of people, which can lead to recruitment challenges.
Data collection method, Delphi panels. The strengths include may provide a method for reaching consensus among appropriate experts and stakeholders on important issues and topics. Anonymous process, when appropriate, reduces the role of ego and interpersonal issues in reaching consensus. Information can be collected remotely, example via email or file sharing software, and at the convenience of the participant. Limitations, lack of universal guidelines for the process. Definitions of expert opinion are variable. No clear standards for acceptable level of consensus, which can make the Delphi process lengthy. Size of expert panel should be considered because it is difficult to achieve consensus among a larger group. Implications for lack of anonymity in the case of modified Delphi panel methods. Potentially high respondent burden if using a lengthy Delphi survey instrument. Can be time consuming and costly. Example, high key opinion leader remuneration costs. Data collection method, observations of patient behavior or events. Example, ethnography studies, which may occur in real world settings and in real time, social media listening, videos. The strengths, opportunity to observe the participants' experience. Maybe low burden for participants because the observation does not require active participation. Potential to observe episodic behavior and signs in a real world context. Observations do not rely on patient or caregiver report. Limitations may be time consuming and logistically cumbersome to execute if conducted in natural settings. Example, study environments may vary across locations. Patient and others' privacy needs to be addressed given patients will be observed in their daily lives. Some concepts and experiences are not observable, can be expensive. Example, equipment if recording behaviors, staff time for observing in real time. Participant behavior may be affected by observer presence. When conducted in a naturalistic settings, variability among settings may affect the reliability and generalizability of the results. Data collection method. Facilitated discussions in organized patient conferences or meetings. Strengths, gain in-depth information on the topic of interest. Efficiency, elicit feedback from multiple participants at one time, can include real-time public polling exercises. Limitations, input is limited to patients who can attend the meeting, which may affect the reliability and generalizability of results. Although panelists speak to the moderator, participants do not interact with each other in the same way that focus group participants do. Representativeness and clinical confirmation of diagnosis may be difficult to determine. See the potential limitations of focus groups. Study materials, study material research protocol. Components, details on how the research will be conducted. Evidence to support the conduct of the study, example, unmet need. Description of all research-related activities and study activities that patients will undergo. Considerations, outline clear research objectives and questions. Identify potential sources of bias. Specify details on target population, including demographics, clinical characteristics, example, phenotype, genotype, disease or condition severity, and other pertinent characteristics, example, geographic representation. Use screening material to ensure the target population of interest is recruited. Specify how data will be prepared for analysis, example, transcription, audio, video recorded, internet data, metadata, archives. Include information regarding projected clinical site enrollment, characteristics, example, geographic location, referral, academic centers versus community centers, to help further characterize the study sample. See Guidance 1 for details regarding considerations for study sampling and representativeness. Identify the number and duration of discussion sessions you plan to conduct. This should be dependent on number of objectives and research questions, demographic and cultural linguistic diversity of the target population, example by age, sex, gender, race, educational level, including number of subgroups, example, disease or condition severity levels, phenotypes, informants, just patients or patients and their caregivers.
Study material. Interview discussion guide. Components. Interviewer, moderator instructions, study instructions, warm-up questions, e.g., questions to establish a connection with the participant. Core topic-related questions. Wrap-up questions, discussion conclusion. Considerations. Use terms participants can understand and avoid technical terms when possible, e.g., choose to use the term shortness of breath rather than dyspnea. Avoid asking leading questions that guide participants to respond with a preferred answer. Avoid asking questions that imply you are casting judgment on a participant's beliefs or choices. Use open-ended questions rather than closed-ended questions where appropriate to elicit spontaneous information from participants. Probing questions may be appropriate based on the direction of the conversation, e.g., if a patient misinterprets the question. Frame questions within the context of a participant's experiences. Avoid questions about abstract or theoretical concepts to the extent possible. Consider eliciting specific data by framing questions using targeted approaches such as diary questions, patients asked to describe a typical day, critical incidents, patient reports best, worst experiences, Free listening, e.g. patients list all symptoms, impacts, and treatments. Ranking, e.g. patients rank importance of symptom, treatment benefit. Study material, training materials, components, detailed coverage of the research protocol contents, consent and assent forms, mock discussion session. Staff can evaluate flow of discussion. Considerations. Train staff using standardized training materials, e.g. training documents, slides, provide refresher training, training on use of technology where applicable. The consent and assent form should contain the appropriate information, i.e. elements of informed consent. Train investigators and staff to present elements of informed consent and assent to prospective study participants and document the corresponding consent and assent. Study material, glossary, components, definition of terminology, considerations. Clearly define key terminology within the text and ensure consistent terminology is used throughout the study documents. Study material, coding dictionary, components, codes, category or concept descriptions, coding structure. Considerations. Outline clear instructions for categorization, including code definitions, instructions, and considerations. Oh, and I forgot some. Under coding dictionary, another component is memo, ideas or thoughts on how codes are derived, and the consideration, derive initial codes from prior knowledge, e.g. natural history, conceptual model, disease model, discussion guide structure, develop a hierarchical organization of main codes and detailed codes. Study material, data analysis plan, components, data to be collected, analytic methods, including coding software, identification of coders and analysts, including credentials, plans for resolving discrepancies among coders and other quality assurance measures, e.g. double coding exercises, description of coding stages, e.g. initial coding, interim checks, including plans for coding dictionary refinement, plans for data visualization, table, figure shells. Considerations. Should be established when planning a research study, i.e. before data collection begins. Determine sample size needed for the study. Specify method for achieving and documenting saturation. Identify and specify appropriate analytic methods for data type. Identify analyses assumptions. Consider approach most appropriate to present data, e.g. tables, figures. Appendix 4, analysis of qualitative data. Section A, data preparation. Qualitative data can be voluminous, so it is important to have a standardized method to analyze and interpret the volume of data in a practical and consistent way. Qualitative data should be prepared before analysis. Preparation can include aggregation or transcription of data from different sources, including in-person interviews or focus groups, recordings, e.g. audio, video, online, internet, e.g. social media, chat room dialogues, metadata, e.g. date of interview, date of interviewer, or name of interviewer, demographic details of respondent, source of field notes, initial ideas of analysis. Section B, data analysis. Table 6 provides considerations for analyzing qualitative data. Note that the steps for data analysis in qualitative studies may be iterative and are not necessarily sequential. Table 6, titled, Steps typically used for data analysis in qualitative studies. Steps, compiling and organizing the data. Description, arrange notes from research and other data collection in useful and standardized order, electronic storage, and computer programs. Step 2, describing and classifying data. Break down compiled data into smaller pieces, reorganize pieces into different groupings and sequences, e.g. codes. Step 3, interpreting data. Use the grouped sequence data to identify the larger meaning of the data. Connect concepts from the data to other evidence, e.g. relevant literature, expert opinion, Evaluate whether no new and important concepts have appeared, i.e. saturation, if applicable to the research objective. Step 4, presenting and visualizing data. Package data in a way that can be easily understood, e.g. text, tables, figures. Transcripts should be analyzed using methods appropriate for characterization and aggregation of study results. There are different approaches to describe and classify qualitative data, some that may involve coding and some that may not. FDA generally recommends that qualitative data are coded for regulatory submission. If a coding approach is selected for analysis, considerations commonly include, but are not limited to, the following. Select the appropriate coding approach for the data of interest. Determine the appropriate level of detail for what is to be coded, e.g., line-by-line coding or select segments of text. Decide what data is relevant enough to be coded. Move methodically to a slightly higher conceptual level initially when coding data. Thank you.
Carefully consider the grammatical form of the coded words to best summarize the basic topic of a passage of qualitative data, e.g. actions versus processes versus nouns. When you have literature, expert input, and appropriate knowledge, a preliminary coding dictionary can be developed. In many instances, codes are driven by the data. The coding dictionary will then evolve as new concepts are identified and emerge from the data. See the table on page 31 for examples. If a coding approach is not selected for analysis, methods that are commonly used include, but are not limited to, the following. Arrange notes about original data in a thematical manner. Ensure your notes precisely cite the original data or precisely locate the places in the database. Implement a procedural check. Take notes and crosswalk them backwards into the original database. It is important to note that regardless of analytic method, you should maintain a methodical analytical procedure to avoid non-systematic and inconsistent judgments. C, saturation. When conducting iterative rounds of interviews, saturation is the point when no new relevant or important information emerges, and collecting additional data will not likely add to the understanding of how patients perceive the concept of interest. Saturation is sometimes used in qualitative research as a principle for discontinuing data collection and/or analysis. However, the use of this principle will be dependent on the research objective. Although there is no set criteria or methodology for how saturation may be evaluated, the steps indicated in the example below is one approach. For example, concepts reported in the first 25% of planned interviews with patients are compared to the next 25% of planned interviews after they are conducted. Both sets of interviews, 50% of the originally planned number, are compared with the next 25% of planned interviews, and subsequently all these interviews, 75%, are compared to the next 25% of interviews. If saturation has not been reached by the initial planned sample size, or more information is needed in certain patient groups in order to fully assess saturation, more interviews may be needed. The goal of the saturation process is to compare the amount of new information that is observed in the first interview set to the second interview set, and so forth. Interviews are typically conducted until saturation is met, and no new important concepts are emerging from the last set of interviews with a diverse group of patients. Table 7 shows a saturation grid example summarizing focus group data. In this example, the researchers identified two symptoms, i.e. symptom A and symptom B, based on literature review and subject matter expert input. In addition, the researchers identified additional symptoms based on the transcripts, i.e., other emergent symptoms. Before assuming that saturation may have been met, it is important to review focus group participant demographics to assess representativeness, i.e., do the focus group participants represent the intended target population as much as possible? See the table on page 32 for this example. D, quality control. To improve quality control of data, consider the following. Thoroughly read all qualitative transcripts and reread the transcripts. Ensure codes are applied consistently to all data and apply quality assurance checks throughout the coding and analysis process. Have multiple coders, e.g., each coder recodes qualitative data more than once to ensure reliability. Examine agreement among multiple coders to avoid inconsistent coding to the extent possible. For example, a subset of data is coded by multiple coders using the same coding framework to examine the extent to which the data is coded in the same way. Document how data are collected and analyzed in a transparent manner, i.e., audit trail, memo to file. Analyze and summarize concepts emerging from the interviews in the order that data are collected, i.e., as interviews are conducted, and display in a table or grid. E, data reporting. Regarding reporting findings from qualitative studies, qualitative data should be presented in a clear manner. It is generally helpful to include participant statements in the participant's own words to represent the qualitative data. Stakeholders should use their best judgment on how to best present and/or report the data.
Appendix 5, Screening and Exit Interview Studies or Survey Studies. Screening or exit interviews and survey instruments may be implemented within the context of a clinical study. They can be helpful in obtaining patient feedback regarding various topics, such as the following: reported changes in symptoms or functioning, worsening, improvement, no change, experienced by patients throughout a trial. Changes may be related to benefits, tolerability, and/or unintended effects. Participant treatment expectations, anticipated and unanticipated symptoms and side effects, viability of proposed dosing regimen, patients' experience with clinical trial participation, for example, in blinded studies, whether they thought they could tell they were on the experimental treatment or not, and why they thought they were on that treatment. Thoughts regarding study procedures and study participation, experience with methods of data collection, for example, user experience with electronic data entry, benefit risk perspectives from the patient or caregiver. Screening or exit interviews and survey instruments can also be conducted with caregivers to obtain similar feedback on some of the above topics if they are observable. Caregivers may have unique perspectives important for medical product development programs. The following are examples of potential strengths associated with conducting screening or exit interviews and survey instruments. Contribute cumulative evidence on demographics, medical history, and aspects of patient experience. Inform initial development or refinement of a clinical outcome assessment in early medical product development through cognitive interviews as part of a mixed method approach. Add greater depth to data in rare diseases or possibly other diseases or conditions with not much patient input, where standalone qualitative studies are less feasible. Obtain participant input on meaningful outcomes and meaningful change by eliciting patient definitions of symptom improvement, stability, or worsening. This topic will be discussed further in other guidances. Provide information on trial integrity. For example, whether blinding to treatment assignment was maintained. Potential limitations of screening or exit interviews and survey instruments include extra burden on site staff, for example, additional operational procedures beyond the clinical study, extra burden for patients or caregivers on top of standard clinical study protocol. For interviews, issues might arise regarding interview scheduling, administration time, and confidentiality. For example, certain sites or countries cannot share participant contact details with third-party vendors who might be conducting the interviews. If screening or exit interviews aren't implemented, FDA generally recommends a trained, neutral, third-party interviewer to conduct the interviews. Interviews should generally be conducted before, that is screening interviews, or after, that is exit interviews. Patients complete the main portion of the clinical study to avoid any potential compromise of study integrity.
Thanks to everybody for listening to this important guidance on patient-focused drug development and what matters most to patients and families. We hope you enjoyed listening and learned a lot. We did, certainly, as we read it. Thank you to the FDA, and thank you to every rare disease patient community.